Welcome to my presentation on computer vision to evaluate field experiments. My name is Andreas Hund. I'm from the crop science group at ETH Zurich, led by Professor Dr. Achim Walter. And here in the background, you can see the field phenotyping platform we are working with. But first of all, I would like to show you which crop we are working on. It's mainly legumes, for example here pea, but also soybean, and we are focusing very strongly also on wheat and really trying to phenotype many different genotypes. Here you can see that we also work on oats, but also in buckwheat, and you can see the pea and the soybean experiments. We are doing most of the work in the platform ourselves, from plowing to sowing. Then we are observing the crop throughout the whole season. You can see that there is a lot of stuff going on, like too much rain, cold, heat stress. We try to follow that with different sensors, for example with drones, as you can see, with this multi-view sensor rig, which I'm going to talk about more. And then we are trying to place that on top of the crops in order to get, for example, 3D information out of them. We are doing intensive ground truthing, as you can see here, the PhD student Olivia Zumsteg, and we are also trying to see what other devices can do, as you have seen the literal here. And the whole team is then going to harvest the seeds for the next season, but we also do combine harvest of the whole plot. So we have a kind of a dual harvest. At the moment we operate 2 of these harvest machines, and then we are going into post-processing. We also operate a weather station, which is important for us, and we are trying to sense the soil to get water content out of the soil. And here you see some prototypes. This is a prototype rover from Caterra with a prototype chlorophyll fluorescence transient device on it from Jülich, and we are evaluating if we can operating that for soybean, for example. We are also part of Emphasis. That means what we try to do is we try to team up with other crop people from other stations doing similar things as we do, simply because at some point one needs bigger data, and that's why I would like to get this information in. And here we have the device which we do. So this is the prototyping which we do. The 3 makers of this device are Norbert Kirch, Gessler, Lukas Roth, and Platel Klutsch. Without them, this device would not exist. And of course, the financing from Achim Walter. What they do is we have 13 cameras completely triggered in parallel. This is very important. And you can see they are focusing all from 3 meter height on one point of the crop. The central camera here has a wider view so that we can get the whole plot on top. We have a global navigation satellite system on board to know where we are, and some other devices. Now, what we try to get out of it is 3 point clouds, but of course also 3D visualizations from different angles. And we do regular calibration for that purpose in order to get that right. From the first sensor head where we did not have this, only 1 camera in this case, you can see that here PhD student of Lukas Roth was benchmarking this or was aligning these images so that we can now really follow almost individual plants, at least in the beginning of the season, throughout the plant. And this data set is actually published here. So the basic philosophy what we do is, or what we have is, we have 3 categories of traits. One is the timing of key stages. This is more or less phenology. I'm coming to that also later. The second is the quantity at defined time points. So for example, what is the start and the end value of a trait, and then those response curves, which are asking the question, how does a crop go from one end to to the other end. That means what happens when there's, for example, a cold spell, as is depicted here. I'm not going too much into detail of this temporal trends. I would like to talk today mainly about how yield components are formed and why it is important to understand that. So what we can do is actually to follow the number of plants. This is fairly simple, but actually in wheat not yet developed. There is spike per plant which you can count. And then something here dashed is the most important parts, namely the florets per spike, the grain weight, which is something we cannot assess from outside. It's very difficult in wheat. To make you understand why this is important to understand these components, I have this graphic set up. So again, we have plants and plant deaths, so the plant emergence and plant deaths. We are not reaching the same density as we actually planted. We have tiller emergence and tiller death. That means not all the shoots make it into a developed ear. And then we have this feature, which is particularly important. It starts at the stage of tillering, which is 21, and it goes roughly until start of stem elongation. And in this stage, the flower is formed. That means the different spikelets within this structure, and this is really a small a few submillimeter scale structure, which is forming the spike. That means everything which is happening until that 31 stage is determining how many of these side branches called spikelets you have in the crop. So it's very important to think about that. That means if we are able to count them here, we are able to know what happens until there. And the second thing, what happens is then the spike is growing, and the growth is very important. So the partitioning of photosynthetic assimilates to the growing spike are the next important part. And this is also where then the florets grow. So it means more florets, getting more resources are leading to bigger spikes. And we would like to, in the end, measure, for example, the volume of the spikes, and I'm coming later to that. And then the last thing is that many of these florets are not only growing, but they are also surviving and set seeds. So we have a high number of seeds, which is then determined somehow into grain filling, and then the final product would be a high grain weight per unit seed. What can we do with phenotyping? We can measure these phenological stages, for example, by following height, start of stem elongation, following the number of tillers, which is of heads or of spikes, can get you an idea about heading, and then maturity can be assessed by following the change of the green color to yellow. Then you can know when the process is finished. So we could theoretically use that phenotyping information to get more information about different genotypes and how they go through the season. So the phenology is heritable. Why is that important? Simply because it is important when the plant is going through sensitive stages, particularly when it is around flowering, for example. So heat around flowering is important trait. And here I depict about a 40 years period at Zurich Reckenholz, where we measured, or Acrometeo station, or the Meteo Swiss station, measured temperatures. And here each of these ticks gets you the proportion of days during that period with at least 1 hour at a particular temperature. So you can see that here frost of -4 or heat of above 31 degrees centigrade doesn't usually happen during stem elongation of wheat, which is very important because it shows us that we can probably also relax a little bit and expand this growing period, for example, to more extremes. So it's kind of something we are considering. To model all that is kind of a sequence of processes which we would like to do. For example, apart from a good experimental design, then we need to have feature extraction. I think that's currently the most important part for phenotyping, to get the information out of images or out of a hyperspectral signal, but then of course also downstream correct for spatial effects in the field, and then very importantly try to model that dynamically and in the end predict yield. So we have a sequence of different steps to model what we try to get out of a product, and I'm focusing now on the first part, namely on the modeling of or the extraction of stuff from images. What I believe is very important, that you have some sort of stereo vision or 3D information from your plot, because this is giving you, for example, height, but it also gives you orientation of organs and helps you to get volume or whatever. Segmentation from images, and also some counts, like for example the famous weed head detection counts. Weed head detection is already done, so if you have kind of an image footprint from 3D information or from a binocular vision, you can now get the area which was measured, and then you can also get the information to what certainty you have a certain certain feature in, for example, spikes. So now we are going into the next iteration of it, namely the segmentation of the canopy into organs. The idea behind it is to go from a global weed head detection data set, which is one of the largest head data sets we have in weed, and we go to the next step where we try to get all the organs segmented. So the basic idea was here put down from colleagues, where they, in the CAPTE unit, where I'm actually at the moment doing a sabbatical, where they were saying, okay, we would like to segment the whole or describe the whole canopy exhaustively. And what we did in this semantic segmentation, which I was guiding in the recent years, was getting the spikes, the leaves, and the stems out of these images. That was a large consortium of 11 institutions gathered around the whole world, a total of more than 60 field experiments, and you can see here quite a large diversity of images which you can see, or which we collected. The diversity of these images, which we then annotated, we did 1,096 images, is actually here summarized in this UMap, where you can see that phenology plays a large role in the first dimension of this dissection. You have, for example, here emergence, and then going through the different vegetative stages to early filling, early senescence, late senescence, and maturity. So this is a transition going from left to right, whilst the different institutions are more stratified in the second dimension, where you can see, for example, down there you have University of Tokyo, and up there it is CIMIT, I guess. So I think this shows that the diversity is immense, and probably also needed to collect this diversity. And then we are looking into classes, so we actually sampled quite a substantial number of different stages. We selected equal frequency of heads, stems, and leaves in the images. Only with the pixel class, the stems and the heads were not so widely visible in the image. But of course, that's an image feature. When we tried to run a first benchmark model, this worked decently well for the heads or the stems, for the heads and the leaves, but not so much for the stems, where you only acquired 40% of the overall intersection over unit. I'm not going to the right graph. And here you can see for the two models which use DeepLab v3 and SegFormer, you can see the parameters. You can also see if we are leaving some regions out, the performance dropped. Excuse me. So going next. This is what the annotation looked like, the labels, which were human-made, so human labeling. And this is kind of the product of the SegFormer model. And I show you now some details in the images. For example, here you can see that actually this is nice, that we are actually not measuring a weed in the field or a weed infestation, but really the weed plants is detected. And also the organs are still decently detected even in quite complex and brownish canopy situation. In the next part of my presentation, I would like to present some work which was done by Jonas Anderegg. He was first also training a semantic segmentation of all the weed organs based on a very small data set or a smaller data set. And I show you the results of this data set now with a small sub study. So he followed the organs, here the stems and the leaves, through time, and also followed the green fraction of these organs. So there was kind of a deep learner and then followed by a shallow learner on color. And what you can see here is that you can now follow these different organs, the stems, for example, through the season, and you can then take the organ which makes most sense, for example, for physiological maturity, which is the stems. So you can take the stem as a measure for physiological maturity, leaving out the other organs, which may have a different dynamics dependent on, for example, if there are leaf diseases present, or also maybe dependent on what the zinc strength or the ear does. So we are just trying to learn how to use these tools because they are very new. But before I go back to this seasonal change, this comes back later, I would like to go now into the fruiting capacity story, where we try to get the spike volume out of images. So by the way, when I talk about ear, spike or head, it's all the same, it's just different expressions of the same thing. So again, you have seen that already. So a large partitioning of photosynthesis to the growing spike is actually something like the fruiting capacity. The overall spike volume at anthesis is set the upper limit of yield. So we'd expect a high yield if you have a large volume. But then we do not know how efficient these plants are setting then seeds. So there can be abortion, there can be problems with grain growth. So the fruiting efficiency is the other measure. That means we need to understand yield formation. Actually, we need both fruiting capacity and efficiency. So fruiting capacity was defined also by Gustavus Slaver. There's a wealth of literature available, and it is said that the higher spike ear dry weight at anthesis is related to a high grain number. So this graph here is not showing this relationship, but the relationship between the spike volume, which we estimated based on scans of individual ears, and the spike weight. And you can see that actually, according to this definition, I would say that also the spike volume itself can be now used as a proxy for this fruiting capacity, as we term it. So fruiting capacity, what is it? It is actually the spike numbers and their volume, which gives then the total spike volume in a crop or in a canopy at anthesis, for example. So this is we term this fruiting capacity. So what we need to do is count ears and measure the size, or count spikes and measure the size. So this is work in progress at the moment. Olivia Zumsteeg is also giving a presentation about that. And what she did, she was tagging individual spikes to get the ground truth and then scanned them. So we have a 3D model, so to say, and also the volume of it. And we use that information, or that scans, also for the experiment of Jonas. And Jonas was doing a shading treatment, and I'm coming to this treatment in detail later, which he kind of took away sunlight. That means there was less photosynthesis for the growing spike in a phase where the spike was intensively growing, and you can see even visually that the shaded were then becoming smaller. I come to all this stuff later. So this is kind of the scanner, but also we did then in the end a seed analysis, the seed size analysis. So what he did here, Jonas, was a shading treatment from the flag leaf, that is when the last leaf is fully developed in the canopy, until heading, that means when the spike, until the time when the spike comes out. That means that was during an intensive phase of growth. And what you can see here, that actually there was a reduction in spikelets, and the spikelets were mainly reduced at the proximal and distal end of the spike. So this is what you can also see in this image. So the lower spikelets and the upper spikelets were underdeveloped simply because there were also lower number of florets developing in that spikes, or there was abortion during that phase, leading to really lower structure, lower volume. You can see that quantitatively here. Actually, I think this is just pixel numbers. It's not something calibrated, but you can see that here you have a reduction in the spike volume. We have two genotypes, a photo weed and a high quality weed. And this is also an important and nice side story here. So from the phenology or point of view, the two genotypes are not remarkably different. For example, they have the same spike volume, they have the same spike size. But what you can see is that they differ for grain weight or grain yield. That means that actually per se they also differ for the fruiting efficiency, with a lower fruiting efficiency for that variety. And that is actually the high protein wheat, which is, or the bread wheat, which is actually putting more emphasis on high protein content. So actually, again, it's not only yield that counts, but it's also the quality, in this case, the protein content. So you can see that there was a reduction in spike volume and also a reduction in the grain yield. And I think that was kind of going in parallel. And you can also see that the grain protein content, so GPC in percent, was going up for both wheat types, of course, more for the bread wheat. And that was really reaching scary high numbers of almost 18%. What you can also see is that the 1,000 kernel weight, that it means related to the individual grain weight, was going up for the fodder wheat, but not for the high quality wheat. So again, this shows that there are different strategies to deal with it. Apparently, the bread wheat was not able to compensate by putting more starch per unit grain weight. And of course, also an interesting story here. So again, coming back to this story of fruiting efficiency, when you assume that everything worked well until the full development of the spike, that means here on the left side, here and there, the spike was theoretically fully developed. It would theoretically set a high number of large grains, you can have still a lot of things going wrong. In the early phase, around flowering, you can have kernel abortion. In the later phase, after this spike number has been secured, you can still have a low grain size. But again, importantly, this happens more or less also in sequence. That means you can deduce from the product, from the end product eventually, also what happened during what phase of this process. Again, what you need to do here is spike counts, spike volumes, but also, of course, all these disruptive harvests. And I would guess it is very important and mandatory to know about protein content and 1000 kernel weight. Now coming back to our phenotyping, that's what we are talking about. Here you can see that the shaded or the control treatment underwent this expected rapid senescence of the green leaf. In this case, we have followed the leaf fraction during the late phase of grade filling. Whilst you can also see that the shaded treatment was having this very long senescence phase. Even with the large images which we took, some leaves were still green. That means what you can see here is that apparently under shading treatment, when these ears were small, the plant apparently did not know what to do with the nitrogen which was left in the leaves. That's my interpretation of the story, and where to put that. So it was kind of having a delayed senescence. Here we summarize all the different processes, the senescence of the ears, or the stems, and the foliage, and we took the midpoint of this senescence dynamics, and you can now see that the genotypes in fact do differ. That means whilst the fodder wheat has almost no delay in the senescence for the ears and the stems, the leaves showed a delay. And if you look at the high quality wheat, you can clearly see that all the different organs showed a delay in senescence. So again, it matters which organ you follow in the canopy, and maybe it also matters how you then interpret the data. And we are just starting to learn how to use. With this, I'm at the end of my presentation. I would like to thank the funders of our work, the whole crop science team, the ETH group of plant pathology with Jordan Anderegg and Radek Zenkel, the breeding partners, the colleagues at Arvelis and Gustavo Slava, and also the Global Wheat Data Sets colleagues. And with this, I also want to thank you for your attention. There's a small video of the FIP if you'd like to watch that, and I'm open for questions.